Hello everyone, my name is Bruno Costa. I come from the University of Minho, and I would like to thank the organizing committee of this Congress for giving me the opportunity to talk to you a little bit on the molecular and cellular hallmarks of brain tumors, with a special focus on their prognostic and therapeutic insights. So starting with a brief introduction on gliomas, these constitute the majority and also the most malignant primary tumors of the CNS. They are a very heterogeneous group of tumors, of which there are two major histological subtypes, namely astrocytoma and oligodendroglioma. These can be divided into different grades of malignancy, from lower grade 2 tumors up to grade 4 highly malignant glioblastoma, and over the years a variety of different molecular alterations have already been described, some of which have prognostic value. Indeed, the most recent 2016 WHO classification of tumors of the CNS reflects a paradigm shift, in which the traditional histology-based glioma diagnostics was replaced with an integrated histological and molecular classification, which allows a more precise tumor categorization. This figure nicely summarizes the major classification criteria currently used for gliomas, and again, this includes obviously histology, but also the status of IDH1 and 2 mutations, the nuclear expression of ATRX, and also the co-deletion of chromosomes 1p and 19q. Together, this integrated diagnosis allows us to differentiate oligodendroglioma, which are always IDH mutant and 1p 19q co-deleted and can be of grade 2 or 3, and then we have different combinations for astrocytoma, depending on IDH mutation status and also on their malignancy grade. Of note, tumors that are located in the midline region of the brain are considered a different entity and typically have mutations in histone H3 lysine 27, and these are called diffuse midline glioma. It is also important to highlight that this additional layer of information at the genetic level is important not only for the diagnosis of these tumors, but also helps in taking therapeutic decisions in the clinics. For example, glioblastoma patients that have an IDH mutant tumor benefit from a therapy that consists of temozolomide in combination with radiotherapy, followed by adjuvant temozolomide, while in the context of patients with an IDH wild-type glioblastoma, particularly for those that are older than 70 years old, it is also important to evaluate the status of MGMT promoter methylation. What happens is that when this MGMT promoter is methylated, the protein is largely not expressed, and as a consequence, the tumors maintain sensitivity to temozolomide. In contrast, in the context of a non-methylated promoter, MGMT will be overexpressed in the tumor, and as a consequence, the tumor cells will be able to repair the temozolomide-mediated DNA damage in the cells. Of course, in this context of temozolomide resistance, the patients are treated only with radiotherapy. So in summary, this highlights the importance of integrating different genetic markers, also to help in taking more refined therapeutic decisions in the clinics. Today I'm going to focus particularly on glioblastoma, or also known as GBM, because this is not only the most frequent but also the most malignant primary brain tumor. Patients have a very poor prognosis with a median survival of approximately 14 months, and unfortunately the treatments are still only palliative at best. When we compare how this situation has evolved in the last 3 decades, we can see that the survival rates for brain cancer have improved only 1%, which is in large contrast with other tumor types or with all other cancers combined, which is around 20%. Looking at the numbers in a different perspective, if we look to the 5-year survival rates, only 1 out of 10 GBM patients will survive after this period, which is very different from children that have leukemia or women diagnosed with breast cancer, with numbers around 90% of 5-year survival rates. So again, it is a very aggressive tumor, and the situation has not changed significantly in the last decades. More recently, various studies suggested that glioblastoma can be further divided into molecular subgroups. As exemplified in this figure, up to 8 different distinct entities of glioblastoma can exist. These are based on different combinations of particular genetic aberrations, and of course, we are not going to go through all of these details, but what I would like to highlight here is that each of these glioblastoma subgroups appears to have a particular clinical presentation. For example, these subgroups appear in particular age groups. The location of the lesions within the brain also appears to be associated with these subtypes. And finally, the prognosis of the patients can be also quite different. We have median overall survivals for some groups that are around 12 months, while in others this can be beyond 36 months. So again, this really highlights how we may be able in the future, based on more refined molecular profiles, to actually better predict what will be the clinical outcome and also the clinical behavior of these tumors. One concept that is very important to keep in mind is the notion of intratumoral heterogeneity, which is actually a hallmark of glioblastoma. So very basically what this means is that within a single lesion, we are able to find very different molecular clones of cancer cells. So when we look at the molecular subtypes that have been previously suggested, here we can talk about, for instance, proneural, proliferative, and mesenchymal subtypes. What we now know, based on single-cell RNA sequencing studies, and this science publication is a paradigmatic example of that, is that we can actually find within a particular tumor the 4 different molecular subtypes that were previously described. So in a way, this challenges our previous notion that a particular patient would have only a particular molecular subtype of glioblastoma, and we now know that what actually happens is that they may have different proportions of cancer cells that reflect these different molecular subtypes. To make things even more complex, we also have tumor extrinsic cellular players that affect glioma pathophysiology. So, in the tumor microenvironment, in addition to the cancer cells per se, we also have a variety of normal cells. This can include, in the context of brain tumors, neurons, oligodendrocytes, astrocytes, microglia, endothelial cells, immune cells, among others. Overall, a variety of cell-cell interactions, either direct or mediated by paracrine signaling loops, can be combined and favor a variety of hallmarks of cancer, including increased tumor growth, invasiveness, and resistance to therapy. Glioblastomas are also compartmentalized in anatomically distinct regions that display morphological and functionally distinct tumor niches. Here are represented the perivascular niche in A, the hypoxic niche in B, and the invasive niche in C. Overall, these niches are relevant for a variety of hallmarks of glioblastoma, such as the maintenance and propagation of glioma stem cells, and also the remarkable invasive nature of these tumors that renders them impossible to be completely resected by neurosurgery. In our lab, we have also been interested in identifying new molecular hallmarks of glioblastoma. One such example relates to OXA9, in which in a variety of studies, we showed that it is a novel oncogenic prognostic biomarker in glioblastoma. In summary, we were able to define the full transcriptome associated with OXA9 in these tumors, meaning that we identified all the genes that are either upregulated or downregulated by this transcription factor in glioblastoma. We identified the cancer hallmarks that are regulated by OXA9, and this relates to its implication in increasing tumor cell viability, invasion and stemness, and also decreasing cell death. We found that OXA9 is associated with an increased resistance to temozolomide chemotherapy. Actually, we identified the downstream molecular mechanisms that are involved in this chemoresistance, which are apparent mediated by BCL2, and we were able to show that combinations of temozolomide with pharmacological inhibitors of BCL2 were able to revert the OXA9-mediated chemo resistance in glioblastoma. Finally, we also showed that OXA9 is able to transform astrocytes into malignant cells, and at the clinical level, we saw that increased levels of OXA9 are associated with a shorter survival of glioblastoma patients. This association with the prognosis was also observed in a variety of mice models. The next goal now, of course, is to identify more druggable molecules that may direct more personalized therapies, and the reason for this is because OXA9 is a transcription factor, which, for obvious reasons, is quite difficult to target. One example of an OXA9 target gene that is potentially more easy to target from a therapeutic perspective was Wnt6, which in our studies we identified as a new prognostic biomarker in glioblastoma. Using a variety of in vitro and in vivo approaches, we were able to prove that Wnt6 mediates increased cell viability, proliferation, invasion, migration, and capacity for self-renewal, while also mediated decreased sensitivity to chemotherapy with temozolomide. At the molecular level, we are able to see that Wnt6 signals through very well-known signaling pathways in glioblastoma, including the Wnt, the RTK, and the AKT pathways, and more importantly, at the clinical level, we are able to prove that Wnt6 high glioblastoma was associated with a much shorter overall survival of patients in independent cohorts. Another question that we are interested in relates to the interaction between glioblastoma cells and other cells that are present in the tumor microenvironment. For example, we recently showed that there are relevant interactions between glioma and normal glial cells that affect the pathophysiology of glioblastoma. In this study, we showed that glioblastoma cells are able to modulate the secretome of normal glial cells in a way that will favor either the go or the grow phenotypic switch of the tumor cells. Actually, we were able to identify which were the particular molecules that mediated these phenotypes, and again, the major dimensions that we evaluated at the levels of the glioblastoma cell involved cell viability, cell death, and migration. In another study, we investigated how mesenchymal stem cells, or MSCs, may affect the pathophysiology of glioblastoma. And the motivation for this was related to the fact that MSCs have been previously suggested to be potentially interesting drug delivery agents for tumors because they present a natural tropism to migrate to tumor sites. However, in our studies, basically we were able to see that the secretome from these MSCs actually promotes a variety of glioblastoma hallmarks, including increased viability, increased migration, proliferation, and also tumor growth in ex vivo models. We also identified the particular proteins and which pathways were enriched in the secretome as a way to be more informative about potential ways that we can act to revert these effects. Actually, our study raises some awareness and caution related to the potential use of MSCs as therapeutic agents for this disease. Globally, our recent studies have focused on molecular and cellular mediators of glioma pathophysiology, which can be either tumor intrinsic or tumor extrinsic, depending on whether they occur in the cancer cells per se or in other cells of the tumor microenvironment. I just showed you examples of OXA9 and Wnt6 and also on the relevance of glial cells and mesenchymal stem cells. Our strategy always explores their functional roles, the clinical value, and how we can explore therapeutic opportunities related to this knowledge. To conclude, I would just like to raise a couple of questions more related to the topic of this Congress. Firstly, how can molecularly distinct gliomas be distinguished with these new imaging approaches that have been developed? Indeed, there are a variety of studies that have already associated particular MRI phenotypes with a variety of molecular alterations that are typical of glioma. In the future, it would be interesting to evaluate how these new approaches can provide better, earlier, non-invasive, and more refined diagnostics. Of course, they would be critical for unresectable tumors, and also it would be interesting to evaluate some potential causal relationships between molecular alterations, and imaging phenotypes. Finally, how can we deal with the remarkable heterogeneity both at the spatial and temporal levels of gliomas? Are these new imaging approaches able to differentiate heterogeneous regions of the tumors? And what would be the clinical implications of these capacities? I would like to conclude by acknowledging all the team members and collaborators and also the funding agencies, and thank you for your attention.